Hello everyone. In this chapter of Life on Planet Earth, we are going to address cell division. The kind of cell division that applies to skin cells, liver cells, pancreatic cells, cheek cells, you name it. Now, as we dive into cell division, there is a statement I want to make up front. The cell cycle in a living organism has to be tightly regulated. The reason why it has to be tightly regulated is because if it's not tightly regulated, we are going to undergo uncontrolled cell division. That uncontrolled cell division leads to cancer. But we can ask, what causes specifically uncontrolled cell division? Well, what causes uncontrolled cell division? It's an alteration of genes that are responsible for controlling when the cells should divide and when the cells should not divide, right? There are two families of genes that are involved in controlling how often the cells should divide or not. We have the proto-oncogenes. The proto-oncogenes, their job in the cell is to stimulate cell division, right? They say cell division should start, right? It's the equivalent of a green light when you are driving, right? An example of such gene is the RAS gene. The second group of genes, it's the tumor suppressor genes. The tumor suppressor genes, they technically tell the cell, stop cell division, stop the cell cycle, right? They cause cell cycle arrest, right? They are basically like a red light when you drive, right? When you reach the red light, you have to stop, right? An example of such gene is P53 gene. Now, what happens is that these groups of genes are tightly controlled and tightly control the cell cycle, right? Now, sometimes though, these genes are not doing what they are supposed to do. For instance, a proto-oncogene can actually be modified in such a way that it's overdoing what it is supposed to do. Imagine that you are driving and that your foot stay locked on the gas pedal. If your foot stay locked on the gas pedal, how do you stop the car? There is no way you are going to stop the car, and the potential outcome is a crash, right? Likewise, when the proto-oncogenes are modified so that they are overdoing what they are supposed to do initially, this leads to cancer, uncontrolled cell division. By the same token, the tumor suppressor genes, when they are inactivated so that rather than staying stop, they are not able to stay stop anymore. To take an example of a car again, imagine that you are driving, you are on a highway, and you are driving 70 miles per hour. You reach out to the gas pedal, not the gas pedal, rather the brake. But in spite of the fact that you are pushing the brake, the car is not stopping. What do you think is going to be the outcome over time? Well, chance that you're going to crash is very high, right? So that's why whenever the tumor suppressor genes get altered in such a way that they are not doing their job anymore, which is to stop cell division, the cell undergo uncontrolled cell division, which is cancer. So I want you to remember upfront that cancer is caused by two sets of genes, two groups of genes that can be modified. One, the proto-oncogenes that can be overactive At that point, we don't call them proto-oncogenes anymore. We call them oncogenes. 2, it's the inactivation of tumor suppressor genes, which means that they are now unable to do the job they are supposed to do in the cell. All right, so I told you earlier that the cell cycle is tightly controlled. In the normal cell cycle, the cell undergoes a certain number of events during the lifespan of the cell until it divides. The first event for a brand new cell is to reach a stage that we call G1. When the cell is in the G1 stage, there is a check point. That checkpoint in G1 is going to verify a number of things. That brand new cell, well, is the size of that brand new cell normal? What normal is depends on the physiology and the anatomy of a given organism, right? But I'm not going to go into the details of that. The cell is going to check if there is proper nutrients to maintain the proper function of the cell. There are growth factors that are going to be checked, and DNA damage is also going to be checked. Is DNA the proper sequence or not? Or is DNA damaged or not, right? If DNA is damaged, that DNA needs to be repaired. If that DNA cannot be repaired, then that cell will have to die, right? So some cells in G1 can enter a resting state. That resting state allows them to actually perform the function for which they were designed to be. For instance, a nerve cell, its job is to receive some information and treat that information and send out the information. That's what a nerve cell is supposed to do. A muscle cell is supposed to contract, right? To allow movement, right? So these cells are going to be in G0 to allow them to do their job. Now, if a signal comes in and tell the cell you need to move on and enter the steps through which at the end of the day the cell is going to divide, then that cell is going to undergo the S phase. That S phase is going to be about DNA synthesis. After DNA synthesis, the cell goes into another stage, that is the G2 stage. During that G2 stage, there is a checkpoint, okay? That is the G2 checkpoint. That G2 checkpoint checks for the size of the cell and also DNA replication. Now, as DNA was synthesized here, was it synthesized properly? Yes or no? If it was synthesized properly, then it makes sense that the cell should move on to the next stage of the cell cycle. If that was not properly done, then the cell needs to make sure that replication is done properly. If the damages are such that this is not sustainable, then that cell will have to die. And we are going to talk about that aspect too. How is the cell dying when there is enough damage to the cell so that that cell cannot be healthy, right? So you have a checkpoint, a second checkpoint here. And finally, you have the M phase. The M phase is the phase during which the cell is actually actively dividing to produce two brand new cells. There, the genetic information has to be distributed equally, right? Your skin cells, for instance, when you have a cut for skin repair, each daughter cell has to receive the exact same amount of genetic information and the same quality of genetic information. So here, for the same amount of genetic information, the spindle assembly checkpoint is going to make sure that the chromosomes are attached to the spindle so that the two daughter cells are each going to inherit the exact same amount of genetic information. If not, then that cell is not going to undergo cell division, and that cell is not going to be healthy, right? Even though cell division occurs, right? And something will have to happen with that cell. All right, so with that said, let's move on and take a look at the type of cell division that is happening in what we call somatic cells. The somatic cells are the cells of the body as opposition to sex cell or reproductive cells. So let me write it down. Somatic cells. Examples of somatic cells are your liver cells, your muscle cells, your skin cells, your nerve cells, the cells throughout your body except for sperms and eggs. All right? So these somatic cells are going to undergo cell division. And why do they undergo cell division? Well, for growth. You remember you were a little child. Now you are likely an adult. Okay? And for you to grow from that little small size of human to that full grown up individual, you had to undergo cell division. All right? And that cell division, by the way, it's called mitosis. Mitosis. Another reason for that cell division to occur is for tissue repair. When you have a damage on your skin, that skin has to be replaced, right? So these dead cells are going to be replaced progressively, right? So that you have a complete repair. So brand new cells have to be produced. How about the development of a zygote? A sperm cell meets an egg cell. At the end of the day, you have fertilization. That fertilization leads to a zygote. That zygote is going to divide into a mass of cell. And that mass of cell that doesn't look anywhere like a human being, over time is going to develop. When we say development, we are talking about development of actually new features that were not there before. For instance, this mass of cell has no limbs, right? But this structure right here has limbs. We can see arms, we can see legs, right? That's development, right? And by the way, the zygote actually multiplies, the cells multiply to form an embryo. An embryo actually is nowhere looking like a future human, right? But a fetus, right, this stage, start looking like a human, right? Even though the organs are still developing, right? And then you have asexual reproduction in some organisms, including protists, including amoeba, right? That reproduce asexually. So you have one cell here and another cell here. Okay? Some organisms reproduce asexually, some do reproduce sexually. Now, we are going to focus here on mitosis, which is asexual reproduction. All right. So, cell reproduction is going to include two very important processes. One is going to be cell growth. The second one is going to be cell division. And these two processes, cell growth and cell division, are tightly regulated. At the end of the day, you have two brand new daughter cells that are 100% genetically identical, technically, right? But we know that nothing is perfect, even in the best machinery, right? Nothing is perfect. Now, as we consider cell division, we want to take a look at what is happening to DNA. Now, historically, when we started looking at cells and cell reproduction, one of the aspects cell biologists spend a lot of time on was to look at DNA, because thinking that DNA is everything about the cell in terms of how the cell functions, how the cell is going to be shaped, and so on and so forth. Well, it was found that DNA is the core information for whatever behavior a cell is going to develop. Therefore, when we studied cell division, we spend a lot of time looking at DNA. So DNA actually is folded into a structure that we call a chromosome. Here we do have a chromosome that is duplicated. So you have one copy of the chromosome here, and you have another copy of the chromosome here, and both are 100% identical. Now, when a chromosome is copied, the two copies are held together by a structure here that is called a centromer. And the two copies of the chromosomes are not called chromosomes, they are called sister chromatids. Sister chromatids mean that the chromosomes that are a copy of each other are connected through a centromer. I want you to remember that. Now, when we look at the sister chromatids are condensed DNA, and that condensed DNA forms loops so that it's so dense that we can see DNA under a microscope, right? Even a light compound microscope, right? But when zooming very much into the fibers that make up that chromosome, we are going to notice that the chromosome is folded and forming loops that can be very long structure, right? Now, zooming in these structures, we are going to see that there is a very interesting organization. So zooming in, we do see here a micrograph of the chromosome. And that micrograph of the chromosome shows some little beads, bead-like structure. These bead-like structures are shown here, represented graphically. These are nucleosomes. These nucleosomes are basically a mix of DNA and proteins. These proteins are called histones, right? So you have several histones that DNA is wrapped around, forming these structures. And these nucleosomes pack up together to form coils, right? Now, all together, DNA and protein, it's chromatin. I want you to remember that. Now, the level of compaction of chromatin is going to define if we can see individual chromosomes under the microscope or not, right? Now, beyond this, we have the histones, the proteins, and the double-stranded DNA molecule. Now, we know DNA is copied. How is DNA copied? That's another story. We are not going to get into that. Now, during the cell cycle, we have two major stages. One is interphase. Two is the M phase. During the interphase, the cell is going to spend about 90% of its lifespan in that phase, the interphase. And that interphase is made up of three subphases. The G1 phase that I already mentioned earlier, during which there is growth, right? And that growth means multiplications of organelles, right? And the cell is going to do what it's scheduled to do. Sometimes the cell can be stuck in G1. At that point, we don't call that stage anymore G1. We call it G0, okay? The cell is going to do what it's supposed to do. All right, so upon receiving a signal that stimulates cell division, the cell is going to enter the S phase. That S stands for synthesis, right? Synthesis of what? Synthesis of DNA. I already mentioned that, right? So DNA replication is going to occur during the S phase, provided that that replication is complete and perfect. The cell is going to enter another stage, that is the G2 stage. At that point, now the genetic information has been copied. The two sister chromatids are held together through the centromer, right? And the cell is going to prepare to enter the M phase, right? So we are going to also have further growth here, okay, and prepare to divide. In the M phase, we are going to have 5 phases, among which 2 phases overlap. Among the 5 phases, we are going to have the prophase of the M phase, then we have the metaphase, then we have the anaphase, and then we have the 2 overlapping phases. The 2 overlapping phases are telophase and cytokinesis. Right? These are the 2 overlapping phases. And now, after that, you have 2 brand new daughter cells that are genetically identical 100%, technically, right? But we know that nothing is perfect. So, when looking at the M phase, we start actually, well, initially, during the interphase, we have a given chromosome, right? That chromosome is going to undergo DNA replication. That DNA replication is going to create 2 sister chromatids that are identical and that are held together through a centromer, right? Eventually, during the M phase, the 2 sister chromatids are going to part ways. Okay? When they part ways, they end up into 2 brand new daughter cells, right? Now, what specifically happens during the interphase? I already mentioned this. You're going to have a set of centrioles here. So you have 2 centrioles that sit in an area that is called the centrosome, right? The genetic information, the cytoplasm contain organelles. The organelles are replicated, right? You have the nuclear envelope that is still completely shaped, right? The genetic information is completely diffuse. You cannot tell apart different chromosomes. You cannot, right? Now here, during the interphase, the cytoplasmic content is going to duplicate. DNA is also going to duplicate, right? So at this point, again, the chromosomes are not visible, right? And we have a pair of centrioles. I already mentioned that. Now, after that, now we're going to see a number of events that are clearly visible under a microscope. Now, the first thing we are going to see, well, not the first thing, but one of the things we are going to see, it's actually after duplication of the centrioles are going to migrate toward opposite direction, right? One set this direction, the other set this direction. And also, we're going to have the mitotic spindle that start forming. These mitotic spindle are nothing but microtubules. These microtubules are going to target the centromers, right? A particular area of the centromers that are called kinetochores. I'm not going to bug you with that, but let me spell it for you, kinetochores. kinetochores. All right. So now, what we are going to see next is that the nuclear envelope right here is going to start getting disorganized. Okay, it gets disorganized so that the mitotic spindle can actually drive toward the centromeres, the kinetochores, so they can attach. So you see here, the nuclear envelope is completely disorganized. The centrioles have moved further apart away from each other, are sitting toward opposite direction, and you can see the microtubules are actually targeting the centromeres. Okay? Then we go to the next phase, the meta phase. Okay? Actually, the phase earlier was the prophase, right? Now, in the meta phase, what happens is that the mitotic spindle that are now attached to centromeres and specifically to the kinetochores are going to pull toward opposite direction. So this one is going to pull this way. This one will pull this way. This is going to happen to each of the genetic information that carries that mitotic spindle. So they're going to pull toward opposite direction. By pulling toward opposite direction, this is going to force the sister chromatids to sit on the equatorial line of the cell in the middle, right? Meta means middle. Phase is the phase of the cell cycle during which the chromosomes, not only are they visible, but also they can sit toward the middle of the cell. Now, what I failed to say earlier, it's that during prophase, the DNA is condensing, and at that point in prophase, we can actually see the chromosomes isolated from each other, right? And you can see here the chromosomes here. Let me go back to interphase. I mean, not interphase, prophase, I'm sorry. You see the chromosomes here. Here in the early interphase, you see the chromosomes that start appearing, showing that the chromosomes are condensing, while here the chromosomes are completely condensed, right? In metaphase, the chromosomes sit in the middle of the cell, okay?